So I'm assistant professor at Scripps in San Diego, and I kind of this would be kind of a tutorially researchy talk. So basically I'll go over time. So I'm a little short that I need to go through. So all right, so EQTLs. So EQTLs, let's just get right into it. I think most of the people know that when Nico was talking, but I'll just briefly go through these things. So if you look at gene expression in a bunch of people, you will start to pick up on patterns like this, that for some specific loci in the genome, people, if you basically group them by that genotype in that location. Sorry.
No worries. Just gonna pop it in. Sweet.
Start from the beginning.
From the beginning.
It's great pleasure to introduce next. So basically there are these patterns that you would pick up on and that you see like, oh, people that carry the A allele tend to have higher expression. And so these are called EQTLs, and there are like a whole range of biological mechanisms that are under them, but like in the most broader sense, you could think of them as two main types, the cis and trans EQTLs. So the cis EQTLs are generally the type or cis-regulation is just like the type of regulation that has something to do with what is on that very physical molecule that that gene is on. So you have two copies of the gene, they're just floating around somewhere, and then if, for example, on here you have some specific thing maybe that makes it better to bind through a certain transcription factor, this specific copy would produce more expression. And as a result, you would see that sort of pattern. Now, alternatively, you could have trans EQTLs or trans effects, which are very important and very common, and they're basically they have same type of effect through diffusible factors. So, for example, one way to think of it is that you have maybe a transcription factor, and this thing helps transcribing this other gene somewhere else in the genome. And maybe if you have a T here, this binds better to that gene, so it increases the expression. And if you have a C, it will be less effective and you have less expression. Now, the main difference is that since this goes through diffusible factors that they're just all over the place, the effects that you see is the same on both copies in this case. Okay, now trans EQTLs are very important. We're not going to talk about them because they're hard to find, but there are people who work on them. So we focus on the cis EQTLs. Now, cis EQTLs are generally found this way. You have a certain gene, you look at common variants around it, plus minus 1 megabase per roughly. That's the type of window people look at because that's generally where you expect to see these sort of local patterns. And then you do a linear regression. The ones that are significant, you call them an EQTL. So, who cares? Now, we had a whole talk about this, so I'm not going to go in detail. But basically, so this is like one of those colorful photos that you tend to see. So it's like a GWAS catalog. Recently, every one of those dots is an association to a disease. The problem with that is most of them we don't know why they're there. They're statistically there, but we don't know why. And one of the ways to justify studies like EQTL is that basically these type of intermediate phenotypes, like gene expression, would help you to give you a step between some high-level disease and a variant or a phenotype. So for example, you could have some sort of GWAS signal like this that says somewhere here in the genome, if you have certain variant, you're more likely to catch celiac disease. Now, that's a very long way from a DNA to celiac disease. Now, if you happen to be lucky and you know that, oh, this exact same part of the genome also regulates some specific gene, then maybe you could use that gene, because we know actually what it does, to kind of go and figure out how this whole thing works. So that's like the whole point of the thing. And they give you this sort of causal relationship as opposed to when you do, for example, differential expression. OK, now, so there have been a lot of consortia working this type of thing. The largest one would be GTEx that me and like a lot of other people here have been part of it in past years. And so GTEx consortium, in short, is a big consortium that has like about 1,000 post-mortem donors. They got around 50 tissues from each of them, and in total about 20,000 samples from different tissues of these people. And then they did whole genome sequencing, RNA sequencing, and a bunch of other assays on all of them to basically just identify these type of regulatory patterns and just general variation in regulation of gene expression in different tissues in human. So, now this type of studies usually look into regulatory variation, genetic variation in regulation from two different ways to look at it. And so cis-EQTLs are the most standard way to look at it, but also we can look at these using allele-specific expression. So allele-specific expression, for those who are not familiar with, are it's like this. So you have a gene. There is some sort of regulation going on behind each of those haplotypes. Now, if you're lucky, you might have some random SNP in the transcribed region of the gene. So for example, here we have a T and a C. And for the sake of discussion, this is functionally very irrelevant. It's just happened to be there, right? Now, if you have those things, if you look at your RNA-seq data, you could basically tease apart which read comes from which copy. So for example, in this case, you see, oh, there are more of these green ones than those red ones. And that suggests that this haplotype, for whatever reason, is more expressed than the other one. So obviously, you would generally want these to be roughly 50-50 for genes. Now, so these two things, they have pros and cons, even though they're kind of taken at the same type of phenomenon. So the pros of looking at ASC is that it's very robust, because in a way, it's like a perfectly controlled experiment. You're looking at two different haplotypes in one single individual or cell or whatever that you're looking at. So all the other environmental factors are accounted for, which is really a complicated issue with gene expression data, because gene expression is designed by our creator to help us basically use our genome where and when it's needed, right? So, so basically, ASC kind of gets that part out of it. And then the other thing about ASC is that because it has this internal control, you could even use it with very small samples. Even one sample can give you insight in what's going on there. And it also teases apart the cis and trans effects, because cis effects are by definition in an allele-specific way, but trans effects are not. So it can help you do that. Now, the downside of that, obviously, is that it's not always available. So it's not always the case that you have SNPs just laying around your transcribed region, specifically if your gene is super conserved, you don't have these type of things in them. And also, the coverage is much lower, because the gene expression comes from the entire gene, but this has to be basically a read overlapping a specific SNP. So now, so the first question is that how these two things are exactly connected. So these are not super new types of data, but and people know that these two things have been showing the same thing for a long time. Have been showing that, but generally has been no clear understanding of, or at least among the a widespread community, that is how do you evaluate the effect of these things and how do they exactly compare to each other? So people have shown that maybe the slope of this thing is correlated with the ratio of, for example, the green ones in here, which we would call the reference ratio. But so we're interested to see okay, how do we actually pin this down? So we did this very mind-blowing modeling here. So I can tell you this doesn't need variational learning. But basically, so our equation says that the expression on one haplotype is going to be some basal expression that is shared between the two haplotypes from whatever things are happening there, times some factor k, which is a positive number, that comes from basically that regulatory point. So there is maybe 5 here. This multiplies it by 10. OK? So that's the idea. Now, and this is kind of gets at the heart of the cis-regulation idea, that you say, hey, there is something that changes the expression, and it does it at one haplotype. It doesn't affect the other haplotype at all. Now, so we were like, OK, how far can we go with this? So the first thing is that, all right, so now let's look at this. So we assume we have one variant. So this is the variant point. You have the red and blue copy of it. And so the red one here is better at inducing expression. And then we see how each of these things would look like in EQTL and expression data. So if you have both of the copies to be of the higher expressed variant, you will have basically both copies producing the same amount of expression, and you have a lot of expression in total. So you will end up here in the EQTL plot and here in the ASC plot. Now, if you're heterozygous, depending on which one is on which one, you would fall on this class or that one. So you show allelic imbalance, meaning that one of your copies is different from the other one. But in the EQTL plot, you always file in the middle, because EQTL doesn't care about the phasing of those things. And obviously, if you have the two small ones, you go again in the middle, and you go there. OK? Now if you write this down, it's easy to show So we cannot find the k in this equation, at least given the normal, usual experiments that we're doing. But it's pretty easy to show that you could find the ratio between the two alleles. So if you have two different alleles, you could just look at the ASC data, get the ratio of the x and y here, and that would give you a fold change between the expression of the two haplotypes. We call this allelic fold change. So it's like fold change, but for alleles within something. Now, if you write that little equation of us for two haplotypes and do a little summing and pushing it around, you could show that basically an EQTL can actually be written in form of a linear regression. So it is actually a linear function as long as you don't log transform your gene expression. And also you could show that the same effect size can be calculated as 2 times the slope of this regression divided by intercept and plus 1. Now, it looks arbitrary, but if you write that down, it's like 3 lines of very basic. Now, if you ask a question, I'm going to go over time.
Okay.
Go.
So just real quick.
Yeah.
EQTL data is not linear with log transform, but in EQTL studies, it's common to do like a rank transformation. Is that still?
So when we did this paper a couple of years ago, we did compare the like a QQ, like quantile normalization to log transformation, and they were working the same. Now, when we did log transform, we were learning it with a nonlinear model that was taking that into account. QQ normalization was working very similar to it, and also the other thing is that for EQTL finding, that's not too important, because actually when the effect size is small, which is where you care and you have low power in identifying them, this actually converges to a linear model. Yeah. All right. So, and basically, we did this, and this is from the GTEx. When we actually calculate these two different things from these two independent type of data, you could see that they actually calculate the exact same numbers. So not the exact same numbers, but I mean that there is no systematic bias. They're actually referring not only correlated, but they're the same numbers. So basically, we are taking at the same phenomenon there. So now, using the AFC is kind of a good way to look into EQTLs and just regulatory variation, not because it actually is a meaningful way to look at it, but it's also because it has good mathematical things to it. So for example, so this is the log AFC. So log AFC between two variants that are identical from regulatory points of view would be 0. If you flip the order of reference and alternative, the size would stay the same. You just get a negative in front of it. And it's additive for multiple alleles. So if you have a variant that actually has more than 2 alleles, you could just write it in additive way. And the same goes for different variants. So if you're considering several EQTLs, as long as they're independently functioning, you could just sum their log allelic fold change. So that's a convenient thing. And it's a pseudometric if you get the absolute value of it, which is convenient for some further down analysis. Now, all right. So we are now we are considering a lot of different ways to extend this thing, which I'm not going to go through, but I just want to touch briefly on it. So for example, now that we have this mechanistic model of how we think about cis-EQTLs, we could, for example, take into account, hey, how would this thing change if we actually take into account the response dosage curves for the binding of this thing? And it turns out that can describe some of the response EQTLs that they change their effect size in different environmental situations by just basically the dynamics of this binding. And then also we are looking at generalization of these to various cases and also comparing it to loss of function variants. And that's interesting because basically now that we have this description, we could analytically compare regulatory variation to loss of function mutation. So loss of function mutation is when you just completely lose one copy of your gene, and that's when you lose half of your dosage. Now here we could actually show how regulatory variants compare to this, and actually treat regulatory variants as like partial loss of function variants and see how they work. Okay, so I'm not going to go through this stuff. I'm going to talk about this part. So how do we generalize this to a case where we have many variants? Some of them we know about, some of them we don't. Now, the reason we were interested in this is the following. So, so this is like a perfect case of a rare disease. So rare diseases are diseases that are individually rare, but actually combined, they're very common diseases and they're very severe. And usually, they tend to be caused by a single mutation, and they're Mendelian, or at least those are the ones that we're considering. And this would be kind of a best case scenario you get for them. So this is from Dan MacArthur's group in Broad. They are basically one of the guys to do this type of stuff. So they have 70 muscular dystrophy samples, or patients actually, and they have all the families. So they have done sequencing of everyone in the family, from trios to however many siblings people had. And basically doing everything that they could do, including RNA analysis, they could resolve about less than half of, or about half of these cases. And this is very usual. Like usually more than half of your rare disease patients, you know clearly something is genetically wrong with them, but you cannot find it. So we had, okay, so I'm not going to go through it, the details of how generally these things work, but basically the idea that we had was that, okay, well, let's look at gene expression, which is not such a brilliant idea. So, and the idea would be kind of roughly like this. You say, hey, for each kid, we just pick every gene. We look at GTEx. We have already looked into about 1,000 people. We just look how much expression you have from that gene in muscle in normal people. And then we see if that kid's gene expression in that point is too far off, right? Now, so the idea is good, but I mean, everyone that has looked at gene expression data would know that this is just not a great idea. The reason is that a random kid in a like a NICU is probably not comparable to a 70-year-old post-mortem GTEx sample, right? So how do we go about this? So the way we try to go about this is an idea that is basically let's just look the proband as both control and the case. And that will be basically looking at the ASC data, because the idea is that we're thinking there is one single mutation that has caused this disease. So it's heterozygous. So if that has a regulatory effect, we should pick it up on the allelic expression. Now, and this is not some idea that we pioneered. People have been looking at it for a while. And so basically, this analysis would go something like this. So you would look at the allelic expression for every gene. So you get a cone shape like this. Most of the stuff are around diagonal. You do your favorite test. So you just peel off some stuff. Everything that is out of it, you say this one has too much imbalance. It's probably a thing. Tests that people use for these type of things is usually binomial or beta binomial. Now, there are a lot of good things about these type of tests and way to go, but the thing that is not good about it is that it entirely ignores the population. Like, it does not take into account anything that we know about sequencing all these other people that we have seen before. So an easy way to think about that is like, okay, well, in GTEx we also have AC data. Maybe we get our patient for each gene, we pick the gene, we go to GTEx, look at ASC for every individual in GTEx, and if our patient is out here and every GTEx individual is there, something is probably odd. Now, that would have been nice if we were not seeing all sorts of weird patterns in ASC data. And the reason that we see, and these are actual real patterns of different genes in GTEx, is that basically humans have a lot of natural regulatory variation, basically the EQTLs, in their genes, that depending on how many of them are there, how strong they are, and what type of LD they have, you could see all sorts of weird patterns. So how do we go about this? So we went back to our rocket science model to see, okay, how can we use our model to basically see what do we expect to see in a population of people. So specifically, let's assume we have a population that they have a bunch of genetic variants. So I have one here, but let's say I have a couple of them. And they have certain different effect sizes and stuff, how this would look like. So we start at the easy case of 1. So in the case of 1, we know that, well, the reference ratio, so the ratio of the reference allele to the total coverage, is going to be the logistic transformation of the log effect size of like log AFC between the two variants. So we could write it for 4 different genotypes. It's 0 if they're homozygous, and it's the log AFC with a minus depending on the phasing on the heterozygous cases. And the frequency of each of these groups in the population is going to be just given by mixing of the alleles. So now from the properties of AFC, we could also just write this for the case of n, which is easy and very convenient to do. It's a simple sum to calculate the reference ratio for everyone and just logistic transform it. And then also you have the probabilities in population that you could calculate. Now, this is good, except that pretty much we have none of these parameters in here. Because for starter, I mean, this would be probably a binomial mixture model with about 3 to the power of n different components to it, n being the number of variants. So that's just not a type of model that you could learn. There are too many parameters in it. We don't have any of that. So we were like, OK, so how can we go about this? So this was the model that we considered, which is a kind of a more likely scenario. So there is a gene. There are many variants or many positions that potentially can affect its expression. We know some m of them, and some of them we don't know about. Or some of them we care about, and some of them we just don't. So how would those equations change? So from the genotype groups, just the number of clusters become less, because we just don't care about these guys. We are only considering 3 to the power of whatever is we want to consider or we know about. So that's easy. Now on the effect size of it, so what is going to be the reference ratio in each of these groups now is going to be the same equation that we had, which takes into account everything we do know. And then everything that we don't know that is under that gray area would basically become a probability sum of what is the expected effect of these things. Now, if there are enough of variants in there, this sum here could be well approximated with the normal distribution with some unknown mean and variance. Now, if we have that, we could basically bring the logistic transformation to the other side of the equation. And then what we get is that basically logit transformed allelic ratio is going to be a normal distribution with a mean that is determined by things we do care about, and a variance that is the variance of the effect of the rest of the things. Now, the good thing about this is that this is a known distribution. This is called logit normal distribution. So distribution goes between 0 and 1. Makes a lot of sense for allelic ratios. And we have it in an analytic form. So basically now if we add the rest of the fluff to the model, that logit normal distribution, taking into account the sequencing noise and other stuff, would boil down to a constrained binomial logit normal distribution. Now, this distribution has a very long name. To my experience, it comes up in some field of science every 10 years. But basically, it's a distribution. It's a binomial distribution where the p in the binomial itself is a logit normal distributed. Now, the thing that is cool about this is that the parameters of this distribution are the average log A of c, and the variance is the variance of the log A of c of the remaining variance. So basically, the mean and variance are both actually immediately interpretable values. So with this model at hand, we basically can fit it to the GTEx data. And if we do this, that model now gives us a set of summary stats that without explicitly knowing what's going on in the genome above that gene, it tells us a summary stats of the average effect that you would see. Now, if we have that, we could plug it to this equation that is basically sum of the expression of two haplotypes, and basically calculate the expected variance of this thing using that distribution that we have. So I didn't write it. This would be a couple of integrals. They're ugly, but they're very simple to do them numerically. So we did that. Now, if we do this on GTEx data, we'll come up with these sort of sentences. Gene X in an average individual is going to be half a fold, for example, different from the average of the population due to natural genetic differences. So that's the type of things we find. And we find these in about 50 tissues and around 15,000 genes. So we did a bunch of benchmarking on this. I'm not going to go through the details, but basically it makes sense in simulation. It also is well correlated with estimates of gene expression heritability. It has good area under the curve and so on and so forth. The things that are cool about it is that it's tissue specific. So for example, here I'm showing the estimates that we have for PAX8. So PAX8 is a transcription factor that causes severe diseases in liver. And if you, in thyroid. And actually, if you look at it, you see that PAX8 does have genetic variation a lot in lung and skin and small intestine. I guess that's this tissue. But for example, when you look at thyroid, it actually does not tolerate genetic variation. So, and then also the other thing that is cool about it is that the estimates that we get are not population specific. So when you compare the estimates we get from the African population compared to Europeans, they're actually very similar. You derive them for different genes because the random ASC SNPs that you find in the data are different between populations, but the actual functional variation is not that different between populations. So that's pretty cool. Okay, so now having that, we're like, okay, no, so we have these estimates. Now, how can we test this in a new patient that we have? So for that, we developed a test. I'm not going through the details, but basically taking into account all the other practical issues that you need to take into account, we'll come up with a test that looks something like this. So this is like the main idea of the test that is like, it's a binomial test, binomial-based test. It has one part that is a binomial, takes into account the type of noise that you have due to the sampling. And it has another part that it takes into account the natural variation that we learned before. And then there is a part in the middle that basically takes into account our model that says, hey, if you have this much variation, this is how it should reflect in your allelic ratios. So it's the mechanistic link between the natural variation and the technical variation. And basically, so what this test at the end would do is that it takes a data like this. For each gene, we'll go to GTEx, look at the variance estimate we had, and individually test it if this is too far off for this specific gene. All right, so we tested this on GTEx data. So this is done by Jonah in Thule's lab. So we ran this on around 500 individuals in GTEx. And so this takes about a second to run per person. And it comes back with around 10 hits in a sample. So there are 10 genes come out that says we think these are outliers. And if you look at them, so here on the x-axis, I'm showing the minor allele frequency going from 10 to the power of minus 2 to 10 to the power of minus 5, so very rare variants. And here is the log ratio of that type of gene will popping out as an outlier. So basically what we see is that the outliers genes we find, they tend to be enriched for genes that carry rare variation. Now, when we look at things that we do know should have a regulatory effect, like stop gains and frame shifts and splice variants, we see that basically anivodot enriches the results by over 100 folds. So it actually picks on the things that we think are having an effect on regulation of gene. Now, one other thing that we did, this is like I added a little part. It's we looked at this in the GTEx V8. So this is like work with Thule's lab and Stephen Montgomery's lab and Alexis Battle's lab at Johns Hopkins that we looked at all of the different things you could look at when you're considering gene expression. So we looked at the allelic imbalance, so that's anivodot. We looked at the outlier gene expression. So this is when you correct for the things you can correct for, and then basically you look if your individual is outside of the pack. And by the way, this is an easier case because we're doing it within the GTEx consortium. It's very different from doing it on a random patient. And then last is that Dirichlet multinomial model that basically gets the leaf cutter estimates of splicing clusters and basically tries to find cases that an individual is having an outlier splicing. So we looked like how these things work. So the first thing we looked at was the same thing. So we are looking at the novel genes that carry novel variants or variants that we only saw them once in NOMAD or twice and stuff like that. So less and less like these are least common and these are the most common variants. And the light test is the total expression, the red is the ASC, and the splicing is black. So we see that all three methods enrich for rare variants. So they actually pick on the stuff that we want. Now, the only catch is that this is when we consider all of the tissues in GTEx. So for an individual, we test every single tissue. We pick those genes that they came out in every single tissue as an outlier, or generally came out as an outlier. And then we see a signal. Now, this is a good luxury, and it's important for analyzing GTEx. But actually, if we have a patient, we only have a blood sample. We're not going to get their brain and liver and a bunch of other things out of them too. So this would be the case when we look at the single tissue. And the first thing you would notice is that you entirely lose the signal that you get from the total gene expression. So it just goes flat into the one. Splicing maintains the result, and AST also does a pretty good job. Actually, best, but probably whatever. They do a good job. So we can actually pick on rare variants that have a functional effect, or in this case, likely functional effect, even when we have a single sample that we are considering. So with that, we go back to, I'm going to soon wrap it up and got 2 more slides. All right, so now we went back to our muscular dystrophy cohort of 70 people. So here I'm showing like one example how this looks like. So this is, let's say, one individual, one of our patients. And these are all the genes, so every dot is a gene, and green is the actual gene that we know is faulty. So we compare these to different tests of allelic imbalance. So we have binomial, which is in a sense gold standard for sensitivity of any allelic imbalance. So it just tests if something is statistically different from half and half. And so binomial test picks over 400 genes, and it does pick the actual causal gene too. Now the problem is that 400 genes are too many to follow up. Now we look at beta binomial test, which kind of looks at what are the genes that are outlier with respect to the rest of the genes, and peels off a little further. Now this only picks around 66 genes. But the problem is that it misses the actual causal gene because it's not so crazy out there, but it's a very conserved gene. So when we look at the ANIVA dot, we get 19 hits in this gene, and the first thing you could notice is that there are some dots that are out there, but there are also some dots that are very in here, and it actually picks on the disease gene. And the reason is that ANIVA dot actually knows that this gene, for example, is likely to have a lot of messy regulation in the normal data too, but this one does not like to be messed with, and that's why it picks it up. So now if we look at all the 30-something samples that we have that we actually know what is wrong with them, this is the type of pattern we see. So if you have God on your team, you would like to have one point here that you report one single outlier, and you have 100%. Now we didn't. So what we get here is that, so in the binomial test, we pick about 80% of the cases. In the rest of the cases, essentially there is no allelic signal. Either the gene is just missing in the data, or it's just in full balance for whatever reason. But then we, on average, we produce a median of 150 genes. In binomial, we produce much less, beta binomial, we produce much less number of genes, sorry, about 50, but we basically lose 2/3 of our hits. And when we look at ANIVA dot, we see that we pick on average 11 genes, and we pick basically pretty much everything that binomial picks up. So it looks pretty good. Now the question is that how we do in the previously diagnosed ones, and we did a lot of things on that, but I anticipated that I will be out of time at this point, so I skipped them. But basically, this is kind of the rough story of what we see. So in the half that we did know what's wrong with them, for most of them, the results we find make sense. So we either pick up the gene, or it's not there, and we don't pick up any other stuff, so it makes sense. For 3 of them, we had conflicts that we are kind of looking into to see what's going on. Now for the rest that we didn't know what's happening, in 12 of them, we found known muscular dystrophy genes that seem to be dysregulated. And out of those, 1 of them we already confirmed that it's a new diagnosis, and 5 of them are still, we're following up. And in the rest of them, we did not find any known MD genes, but we found other genes that are there. And in one case, at least, we found someone else in another hospital that seemed to have something wrong with that gene. So we're following up with that too. But with that, I would like to wrap up the talk. So I talked about AFC, which is like a very, very embarrassingly simple way to think about regulation, but somehow it's strange to me why people just looked at all sorts of strange things when they're looking at EQTL data. So it's this. EF haplotype is K times EB. And then we could basically, we expanded this to actually estimate how much is the natural variation in the population. The thing that I think is cool about ANIVA is that it actually estimates the variation in the gene expression, the genetic part of it, in an absolute scale. So it actually tells you it changed half a fold, which is not something, for example, you can get from heritability studies. And then we basically developed a test to kind of test regulatory variation in rare disease patients. Now, I think the thing that I like to emphasize here is I think it's actually very good. Like nowadays, there is like a huge emphasis on like models that you don't need to think, you just throw them, let the data speak and so on, and that's cool, great. But also it's actually very good sometimes to just use the basic principles that you know and expand them, good old school way, to as far as it can go and see if things are the way you expect them. Because that helps you to ask more complex questions. For example, some of these questions that I answered here, there's no way you could just answer them with a classifier because those specific things we are interested in are not directly in the data. The second thing is that it actually helps you to sharpen your questions. So actually, when you do this thing, you realize, oh, I expect based on our current understanding that this should look like that, but it doesn't. And then you can ask more smart questions. OK, with that, I would like to thank everyone. So there have been a lot of people. I'm not going to go through it, but basically Tuli's lab that I did a big part of this work at her lab, and a lot of people in her lab that has been helping. Dan MacArthur's group in Broad, and people at NIH who actually gave us the samples, and Haki at the University of Chicago that helped us with the heritability estimates, and GTEx Consortium for the data and actually work on the rare variants. Thank you.